Good morning, good afternoon. It is a special occasion to be here today since it is to commemorate our past mentor, Frank Austen. And since I'm here today, not only as one of his several postdocs, but also as a president of the European Mast and Basophil Research Network. And since I'm here with my colleague, Dr. Marc Rothenberg, also a previous student at Frank's lab, and there we met many years ago. And is here not only being an ex-student of Frank, but as a president of the International Eosinophil Society. And he will soon introduce to you a partnership between our two societies. If I might have the next slide, please. I would like to introduce you to our present EMBRN Council. We are a small society. I am the president, and you might note that we have also an officer who is from United States. Next slide, please. Because indeed, even if our name quotes European Muscle and Basophil Research Network, we start as Europeans, but we are open to anybody in the world whose interest is on muscle and eosinophil biology and basophil biology, and on muscle and basophil-driven diseases. So during the years, our society was founded in 2011. We have received membership from not only outside Europe, but really on all the world continent. And I have to say that the society is growing, and that we have in our members the leaders in the muscle and basophil research. Next slide, please. I would like, I'm brief because we have a very important meeting today and a lot of science to listen to. Would like you to save the date. We have our next meeting in Toulouse, France, May 29th, 31st. And our website is open, and you can see that you can already submit abstracts. Thank you.
Thank you, Francesca. It is indeed my honor to be here today to first introduce you to the International Eosinophil Society, also known as IES. IES is an organization of scientists and clinicians interested in the eosinophil. The society sponsors biennial meetings, monthly webinars, and other educational programs for its over than 300 members. We aim to review new information about the eosinophil and its role in health and disease. I would encourage you to consider joining the IES, and you can use this QR code to learn more about that. Next slide. I wanted to point out, as Dr. Levi-Schaffer mentioned, we are now forming a partnership with the European Mast and Basophil Research Network. And there's an opportunity to become dual members in both societies by becoming a member of either one alone. Please seize this opportunity to connect, learn, and lead this dynamic group of immune system researchers. Next slide. Please also hold the date for our next biennial meeting. This will be the 13th biennial meeting. We're looking forward to a very exciting program, which is already underway in terms of its planning. This will be in Montpellier in July 7th to 11th in 2025. Next slide. Next webinar that we'll be hosting will be March 20th. And this webinar will be focused on eosinophil imaging. We have a strong lineup of international speakers, and I hope that you'll also consider attending this very exciting webinar that we're planning. I'd like to thank our Corporate Advisory Council, which includes Platinum members, AstraZeneca, Gold members, Allakos, Amgen, GSK, Sanofi, and Regeneron, as well as other Bronze members. Thank you for your sponsorship of this society and the webinar series. I'd like to now introduce our moderator for today, Professor Peter Weller. Dr. Weller is the William Bosworth Castle Professor of Medicine at the Harvard Medical School. He is Chief of Allergy and Immunology in the Inflammation Branch, at Emeritus Chief of Infectious Disease, and the Vice Chair of Research in the Department of Medicine at the Beth Israel Deaconess Medical Center at the Harvard Medical School. Thank you very much today, Peter, for joining us as the moderator.
Thank you very much, Mark. It's my pleasure to serve as moderator for this very exciting session. I would encourage everybody to ask questions after each of the presentations. So use the Q&A button to ask those questions. I will then present those questions to the speakers. The chat function is turned off. All attendees will be muted, and as I mentioned, the chat function will be disabled. This webinar is being recorded and will be posted on the IES website. For those interested in using closed captioning, that can be done by clicking the Show Captions button. Our speaker lineup is illustrated here. Dr. Boyce will initially make a presentation in memoriam for K. Frank Austen, tracing his history and achievements. Francesca Levi-Schaffer will follow with a presentation on mast cells and eosinophils. What do they really have to say to each other? Marc Rothenberg will address eosinophil advances following K. Frank Austen's mentorship. Jenny Hallgren Martinsson will discuss inflammation-induced mast cells in the lung, and then I will come back with some closing remarks at the end. So with that, we turn over to Josh Boyce. Josh Boyce is the Albert Scheffer Professor of Medicine and Pediatrics at Harvard Medical School, Chief of the Division of Allergy and Clinical Immunology at the Brigham and Women's Hospital in Boston, which is one of the largest and most successful research and training programs in this specialty. And Josh is going to share with us his comments on K. Frank Austen's history and achievements. So Josh, please.
All right. And this slideshow. Okay. All right. Well, good morning and good afternoon. And Peter, thank you so much for the nice introduction. I'm very honored to give this presentation commemorating Frank's history and achievements. He was, of course, a giant in our field. His impact will be felt forever, and he was a mentor to many of us on this call. He was born in Akron, Ohio in 1928, and in the summer between his senior year in high school and first year in college, he developed polio. And this experience profoundly impacted Frank, made him become a more serious student and determined to enter the field of medicine, while it also delayed his matriculation to Amherst College, where he graduated in 1950. And here we see him on his graduation day with his beaming mother looking on. From Amherst, Frank went on to the Harvard Medical School, where he graduated in 1954 with honors. And while a medical student, he did a rotation at Mass General in rheumatology with Walter Bauer, who was then the chief of medicine at that institution. And Dr. Bauer became the first of several key mentors who would shape Frank's subsequent career path. Bauer got Frank interested in inflammation and rheumatic disease, and Frank chose to do his residency at Mass General. And here we see Frank as chief resident seated next to Dr. Bauer in the front row of Mass General's legendary Ether Dome. From Mass General, Frank went on to the Walter Reed Army Hospital, where Elmer Becker, another key mentor, gave him his first exposure to serious bench science, fueling his interest in immunology. From there, Frank sought a postdoctoral fellowship with Walter Brocklehurst at the National Institute of Medical Research in London. Brocklehurst had discovered a slow-reacting substance emanating from the lungs of anaphylaxed guinea pigs that constricted airway smooth muscle at exceedingly low concentrations. And this substance, of course, turned out to be the cystinyl leukotrienes. And Frank would spend the rest of his career studying mast cell biology, leukotriene function, and mechanisms of hypersensitivity. From London, Frank returned to Boston, where he worked for 2 years as the unit chief of pulmonary at Mass General, before Robert Ebert, pictured here to Frank's left, who was the dean of Harvard Medical School, recruited him across town to become the first physician-in-chief in what was then known as the Robert Breck Brigham Hospital for Incurables. At age 38, Frank became the chairman of a newly established Department of Medicine at that institution. And Frank's tenure at the Brigham was legendary. He not only put the institution on the map in terms of academic medicine, but he also forged the merger between the Robert Breck Brigham and 2 adjacent hospitals, the Peter Bent Brigham and the Boston Hospital for Women, forming what is now known as the Brigham and Women's Hospital, which, as Peter said, is an academic powerhouse. This photograph shows Frank with his first lab group that he brought with him from Mass General over to the Brigham. And you will probably recognize several people in this photograph who went on to become leaders in our specialty, including Martin Valentine, Alan Kaplan, and others. Frank presided over a department at the Brigham until that department became part of a larger department of medicine as a division of rheumatology, immunology, and allergy. This occurred in 1995. And this was the first time that the word allergy actually appeared in the division's masthead. And this was very important to Frank and his colleague, Albert Scheffer. This division existed as an intact unit until 2019, when it birthed 2 separate divisions that currently exist at the Brigham Hospital, one in inflammation, rheumatology, and immunity, the other in allergy and clinical immunology. And none of this would have been possible without Frank's immense visionary leadership skills as an administrator. Frank prioritized the training of physicians and scientists. When he arrived at the Robert Breck Brigham, there was no training program in immunology, and he quickly obtained 2 NIH training grants that both exist to this day, one for the training of rheumatologists and the other for the training of allergists. Through these efforts, Frank trained over 200 individuals who went on to become full professors at U.S. medical schools or equivalent rank overseas. These included 4 people who would become chairs of medicine, 4 who would become presidents of the quadruple AI, 3 who would become laboratory chiefs at the National Institute of Allergy and Infectious Disease, and the leaders of the largest and most successful asthma research programs in the United Kingdom. And of course, all of these distinguished scientists went on to have their own mentoring and scientific legacies, which expands Frank's influence and amplifies it logarithmically and in perpetuity. It is only fitting that Frank and his dear friend Albert Scheffer were the joint recipients of the inaugural Mentoring Award from the Quadruple AI in 2013, recognizing their immense contribution to our field. Frank, of course, was a scientific giant. We all know this. He published 590 original studies and over 150 review articles across 8 different decades. His work was continuously funded by the National Institutes of Health for decades, and in fact, Frank's grants were never turned down for funding. He made seminal contributions to the mast cell area, the leukotriene area, the complement cascade, and the biology of the bradykinin system, all of which ultimately led to the development of groundbreaking therapeutics for patients suffering from inflammatory diseases. He was recognized by membership in several honorific societies, including the National Academy of Science, the American Academy of Arts and Sciences, the American Association of Physicians, and the Royal Society in the United Kingdom, where he not only became the first allergist so recognized, but also signed the signature book that had been signed by Sir Charles Darwin and Sir Isaac Newton. Frank received multiple prizes and awards for his research, including the Waterford Prize for his work in the leukotriene area, the Kober Medal from the American Association of Physicians, the Warren Alpert Prize from the Harvard Medical School, and he also has an endowed chair in his name at Harvard. Frank's stature as a scientist and an academic leader was recognized well beyond the walls of the Brigham and the Harvard Medical School. In 1977, he was president of the American Association of Immunologists, and we see in the left-hand photograph Frank posing with the board of that organization, which at the time included a very young Anthony Fauci to his right. And on the right-hand photograph, we see Frank pictured with the board of the then AAAI, of which he was president in 1981, and the board included a younger Levi-Schaffer in the back row. Frank also served as the president of the American Association of Physicians, which signifies his stature extending well beyond allergy and clinical immunology. But for all of Frank's scientific and professional achievements, nothing made him happier or prouder than his family. Frank and Joyce were married for 64 years. On the left, we see them pictured on their wedding day. On the right, we see them just a couple of years later with their 4 grown children, some of their grandchildren, and significant others. They had 8 grandchildren, and they have 3 great-grandchildren and counting. And these people were truly Frank's pride and joy. He would regale us constantly with tales of his exploits with his family, including legendary ski trips, mountain biking trips that occasionally necessitated a visit to the orthopedic surgeon, gardening, hiking, and later in life, an appreciation for fine craft beer, a pleasure bestowed on him by his beerhead grandsons. The last few months of Frank's life were challenging in terms of his health, but Frank was always a fighter, and he rallied in time to attend a wonderful celebration in honor of his 95th birthday last March. And this event was attended by many of his former trainees, his colleagues, his friends, and of course, his family. And Frank could not have been more moved or delighted at this event. In summary, Frank was a giant in our field and a highly successful family man. We could all hope to be so lucky. And I personally feel very fortunate to have been one of his many mentees and to call him my friend. I'm sure many of you feel the same way. God bless you, Frank. You will never be forgotten. Thank you for everything you've done for all of us.
Thank you very much for that recapitulation of Frank's enormous impact on our field and for many of us who had the opportunity to train with him. What I would like to do now is turn to an introduction of Francesca Levi-Schaffer, who will be talking about mast cells and eosinophils, what they really have to say to each other. Francesca is a professor in the Institute of Drug Research, the School of Pharmacy, Faculty of Medicine of the Hebrew University of Jerusalem. She holds the Isaac and Myrna K. Chair in Immunopharmacology. She is the president-elect of the International Union of Pharmacology and Clinical Pharmacology and an honorary fellow of the Royal College of Medicine. She's the current president of the European Mast Cell and Basophil Research Network, and she will be addressing us on the topic of mast cells and eosinophils, what they really have to say to each other. Francesca, please.
Thank you. Thank you very much, Peter, for your wonderful introduction. 1 second, yes. I would like to start by mentioning 2 things about Frank. The first one is personal, and it is the biggest compliment that he gave me when I was leaving his laboratory to go back to Israel. And he said, "Francesca, I love you. You prove to be really like a male scientist." And I took it really, and I'm taking it today as a compliment that he gave me. And the second mention is a quote by Arthur Ward that what he wrote about teachers. The good teacher explains, the superior teacher demonstrates, and the great teacher inspires. Thank you, Frank, for being my great teacher. So, yeah, the question is what muscles have, and the eosinophil have really to say to each other. And I've been asking myself since a long time, being a pharmacology or better immunopharmacology, and my research goal has been to try to find maybe shared targets for allergy, but also for several other diseases in which muscle are often accompanied by eosinophils. This slide is not to tell you what are muscle and what are eosinophils. You all know. However, is there because an early observation that I started to do when I was with Frank was that indeed muscle are long-lived tissue cells, and activation doesn't turn them down. On the other hand, induces the de novo synthesis of the preformed mediator and very quickly their ability to be reactivated. And that foster our observations that in several allergic tissues, let me call allergic, they are not allergic to the tissue, but of course what is happening is there, such as in asthma, such as in nasal polyposis, such as in the skin in atopic dermatitis, you see muscle often nearby eosinophils and even in contact with them. So we started to research what they have to say to each other. And we started with release mediators and the receptor. For just as an example, we show that eosinophil contains stem cell factor. Stem cell factor is a cardinal differentiation, survival, proliferation factor for muscles. And indeed, they can induce muscle survival binding to c-KIT. Vice versa, we found the tryptase, that is a quite specific enzyme contained in the granule of muscle, can activate eosinophil via binding to part2 to produce pro-inflammatory cytokines. On the other hand, I was, and I am as a pharmacology, very interested in cell-cell contact. Maybe I should say as an immunologist, because we know that is a more precise way that immune cells talk to each other. And that brought us to find an activating couple, CD48 on the muscle and 2B4 or CD244 on the eosinophil, but also to observe that both muscle and eosinophil have on their membrane inhibitory receptor. And I'm going to talk to you also about that. Now, where are we with therapy? I have to say we are in a very good place. Why? Because we have still the older strategies that work in most of moderate and mild patients, and we have a bench of new strategies coming from the biologics, the new era of the biologics, some already approved, some in preclinical or early clinical science. The way I think we should tackle muscle and eosinophil, if we want to tackle them down, and I think we do in allergy, at least at the beginning, is by muscle silencing. Muscle as a primary movement of allergy, and the best way, according to me, and what I learned with Frank, is not to kill them, but to silence them. And we just wrote a review with the group of Marcus Maurer. The title is, as you see, muscle silencing. And muscle silencing from my lab has been to try to stop the activating receptor CD48 that I say is present of muscle, but also in eosinophil, or by activating with agonistic antibody the inhibitory receptors. Now, so how do I interpret the allergic effector unit? Is the good, the bad, the ugly, or the ugly, the bad, and the good? Let's see. So first of all, eosinophil, where do they come from in allergy? Usually they come immediately after muscle activation from the peripheral blood, and you see how muscles are friendly with them. They are protruding pseudopulse and bringing them close. This takes few seconds, and the physical contact lasts minutes. And during the physical contact, CD48 and 2B4 that are CD2 family members of receptors, one is a GPI and the other one is SLAM related, interact to cause not only the physical contact, but the induced cell survival, eosinophil stay alive, not just only because of the release GM-CSF from muscles. They can release and induce their survival via their receptor, but by this 2B4 CD48 interaction that is not inhibited by steroids, while GM-CSF, it is inhibited. And we have a physical induced cell activation where muscle, even in the absence of IgE and the allergen, can secrete tryptase, histamine, and eosinophil EPO. And we have, again, just because of the physical contact phosphorylation of LAT, of SIC, and also release of cytokines. To show how CD48 is important, we also stain various allergic tissue, both for CD48 associated with eosinophil or muscle, and of the allergic effector unit, and we found it. At this point, however, I started to be interested, however, or in addition, to the concept of resolution of inflammation that I learned by at Harvard, staying with Bruce Levy and Charles Sirhan. And we all know that allergic inflammation is a disease, a chronic disease, characterized by the absence of resolution, but rather by remission phases. And the resolution of inflammation, just to remind you, is an active process regulated by the release of specialized pro-resolving lipid mediators such as lipoxin, resolvin, protectin, maresin, that induce the accumulated granulocyte to die and to be efferocytoted by the macrophages. So we started to relook at the allergic effector unit, and especially when muscle are activated, and notice that the 72 hours activation of muscle started to induce a decrease of survival of eosinophil, and also a decrease in their release of EPX. We look at CD48 as our main activating receptor, and we notice that both on muscle and on the eosinophil, in the allergic effector unit, that is the granata red, is much more expressed, stay high until 24 hours, but then it declines, the same for eosinophil. So indicating some movement towards what I call the good, the finishing, or maybe of this pro-inflammatory activity muscle have with the eosinophils. But we were curious also about the inhibitor receptor. Our inhibitor receptor, I call it, that is CD300A. It belongs to the immunoglobulin superfamily. It has 4 inhibitory sequences, ITIM. Most interesting, it binds to phosphatidylserine and phosphatidylcholine that you know is expressed on the apoptotic cell and also activated cell. So it's very intriguing. And indeed, we saw that if we co-culture wild-type muscles with eosinophil for 72 hours with eosinophils that are apoptotic, we have a decrease in muscle activation as measured by LAMP1. But if we get the BMNC from CD308 knockout mice, this decrease in muscle activation doesn't happen. Then we look at typical allergic disease, atopic dermatitis, and in collaboration with Harry Alenius and Bernard Home, we look at 150 patients, and is the skin of lesional and the non-lesional or normal skin, and we could see that this gene, the CD300A, is enhanced in lesional skin, as well the expression in the skin, the protein expression of this receptor, and is mostly associated with an increase in eosinophil, mast, and macrophages. If you induce a murine model of atopic dermatitis by stripping the skin and putting of albumin as saurus, you can notice that in the absence of CD300A, the skin is much more inflamed, is thicker, dermis and epidermis, indicating that CD300A regulates the pro-inflammatory feature in the case of atopic dermatitis. In a model of allergic peritonitis with of albumin and alum, we followed the expression of CD300A, and we saw that indeed it goes up on wild-type cells in the peritoneal lavage with the inflammation, so with the total number of cells to go down at day 144 when we have resolution. However, if we do the same experiment in knockout mice for CD300A, we don't get to the full resolution as measured by total cell numbers. Please note that CD300A especially is increasing eosinophil, mast, and macrophages. And what we found that is eosinophil. Eosinophil don't clear up in this model, even though the mouse lacking CD300A synthesize lipoxin A4 and express the receptor for this pro-resolving lipid mediator. Per Giorgio Puzzovio, doing his PhD in my lab, decided to inject an over number of muscles, so overshoot muscle in peritoneal cavity of C57 mice that don't resolve at the 6th day, and induce a not significant, but a trend of resolution where he injected the muscle. You see here, eosinophil number decrease. And you could see that this mice has having the peritoneal lavage resolving D1. That is another pro-resolving lipid mediator. But the greater result he get when he took 4 sources, different sources of muscles, so the cord blood muscles, foreskin muscles, nasal polyp-derived muscle, and LAT2, that is a muscle line, activate them in vitro with Ig, anti-Ig, and he saw the production due to the 15 lipoxygenase of resolving D1. And you can see that the production in all the cases of muscle, connective tissue, or mucosa is quite high, is 4,000 picogram per ml. In the allergic effector unit, what happens? There is a very early production of the resolving D1, and this stays constant until when we measure, that is 72 hours. Importantly, we incubated the eosinophil activated with SPAF or muscle activated with Ig, anti-Ig, the eosinophil from peripheral blood, and we could see the pharmacological concentration of resolving D1 indeed decreases their activation. Therefore, I would like to share what I think we found in our recent research on what muscle have to say to the eosinophil. I would like to hypothesize, based on the results, that muscle can produce pro-resolving lipid mediators, which are only resolving D1, and orchestrate not only the initiation, but also the resolution of allergic inflammation. They are really potent cells. And this resolution can be modulated by the inhibitor receptor, example CD300A, and by the production of the lipid mediators, and possibly by their crosstalk. So in my presentation, I think I show you that the allergic effector unit, even when muscles are not activated at the beginning as the bad or the ugly, is a pro-inflammatory crosstalk. But I think I also convince you that inhibitory receptors such as 3300A, together with the potential of muscle to produce upon immunological activation, Ig-mediated resolving, can induce in a time-dependent fashion a homeostatic resolution, or at least less flare in allergic response. I would like to thank, I don't know where it disappear, my student, all my students for the contribution, my present and past colleagues, and thank you for listening.
Francesca, thank you very much. I would remind everybody that each of these presentations is available for questions. If you have questions, use the Q&A button, enter your question, and I will share them with each of our speakers. I might start with a question for Francesca. The eosinophil mast cell axis, very intriguing. And you mentioned in one of your slides the wealth of modalities now that are available that may be targeted toward eosinophils, towards mast cell eosinophil related allergic inflammation. To your knowledge, if one looks at the anti-IL-5 directed therapies, are there any implications for their effects on mast cell activation?
Yes, first of all, thank you for the question, and sorry, somehow I missed the last slide with the most important people who are my students. Somehow, I don't know why. I answer your question. Yes, it was noticed that in patients treated with anti-IL-5, there seems to be one of the best drugs for eosinophil-mediated allergic diseases. Also, there is less muscle eosinophil interactions. So yes, it helps where steroids seem not to help. Yes.
Following up on that, here's a question from Bob Lewis. What happens to the time course of mast cell activation, both in vitro and in vivo, if you knock down or knock out 15LO?
Thank you very much. We would like to do that, and we were intrigued by the fact that in time, the 15LO remains high. It's not regulated like COX-2 or LOX-5.
Very good. Another question's just come in. Professor, can targeting the allergy effector unit help with patients with multiple allergies?
I think so, because the allergic effector unit is pro-inflammatory independently on what is IgE directed to and of the allergen. So it should help independently of the multiple allergies and independently of where the muscles are. They can be connective tissues, they become mucosal, and nevertheless, they have this interaction with the eosinophils.
Very good. Thank you. I think in order to stay on time, thank you, Francesca, very much for this presentation. And I would like to move on then to introduce Marc Rothenberg. Marc is going to talk to us about eosinophil advances following K. Frank Austen's mentorship. Marc is the Director of Allergy and Immunology at Cincinnati Children's Hospital Medical Center, a professor at the University of Cincinnati College of Medicine, founder and director of the Cincinnati Center for Eosinophilic Disorders, founder and director of the NIH's Consortium of Eosinophilic GI Disease Researchers, and the incumbent recipient of the Bunning Chair of Allergy and Immunology. So Marc, please consider it eosinophil advances following K. Frank Austen's mentorship. And
Thank
again,
you.
questions, if anybody has questions, please enter them in the Q&A. So Marc, please.
Can you see that all right? Peter, is that okay?
We just have the introductory slide.
Yeah. You don't see the one that says these slides? Not yet.
Try sharing the slides again, Marc.
Does that work?
That's working, looking better. Good.
Excellent. Terrific. It's my true honor to be here, as I stated before. What I'd like to do today is talk about my pursuit of understanding eosinophilia and really focus in on the types of research that was advanced based on the wisdom and guidance that K. Frank gave to me. Frank was my PhD advisor, and he subsequently was my long-term mentor. And I'd say, similar to what Josh mentioned, that our relationship did grow into a friendship. This picture shows me as a PhD student on the left. On the right, 30 years later. What is interesting is that Frank looks the same in both pictures, pretty much. In the photo on the right, I am Frank's age in the picture on the left. Now, if you are a keen observer, you will notice that I'm actually wearing the same tie in both pictures. Now, Frank didn't just talk the talk. Frank walked the walk. And what I mean by that is that he devoted his time and energy to truly meaningful and impactful science and providing sage advice. This is illustrated in this photo when he made the effort to visit Cincinnati Children's Hospital for several days to serve as our external scientific advisor. Another example of Frank's dedication and zest for life was when he accepted a speaking engagement in Israel at the age of nearly 90. He told me that he did this in large part to learn about Israel and to spend time with my family and I. One of Frank's pastimes I learned was estimating the time of the sunset. This photo captures the bet that we had on the Tel Aviv beach when he bet us at the time that the sun would set. As usual, Frank proved to be the winner. My research with KFA started with the fundamental observation shown here. This was that the peripheral blood eosinophil was in fact a long-lived cell. This was contrary to the conventional wisdom at the time, which had indicated that eosinophils died soon after entering an inflammatory site. KFA's response to this initial finding was shown here. Don't answer a question that I didn't ask, because this experiment was actually meant to be a negative control for another experiment that he asked about, which was related to basophils. Nevertheless, Frank let me pursue this in his lab, and eventually proving the theory that provided the basis for a new paradigm that eosinophils were long-lived cells that appeared to be tissue-resident cells, not just peripheral blood cells. In the early findings of this set of experiments, we speculated that eosinophils were, that this system was relevant to tissue eosinophils, as these long-lived cultured eosinophils had acquired new phenotypic post-mitotic properties related to their hypodensity, enhanced leukotriene production, and cytotoxic activity. We soon went on to characterize the eosinophil poietins, and proved that one of them, IL-5, was contributing to human disease. In the shown experiments on the left, we first demonstrated a dose-response increase in the eosinophil survival activity of serum of patients with hypereosinophilic syndrome. We then showed that an antibody against IL-5 blocked this activity. These findings were indeed driven by KFA's principle that if it doesn't dose response, it's not science. Based on these collective findings of my research and others, we proposed a model for the regulation of eosinophilia in this New England Journal article review. Of note, we emphasized the importance of tissue chemoattraction signals in addition to the role of IL-5 primarily in the bone marrow. Our research on this subject started off when we had cloned the eotaxin gene, initially in guinea pigs, based on an N-terminal sequence provided by Tim Williams' group. The figure shown here is an initial observation of the pattern of the eotaxin expression in the guinea pig based on northern blot analysis. The surprising point about this data was the interesting constitutive expression of eotaxin in the lung and intestine, the two tissues that harbored eosinophils under homeostatic conditions in guinea pigs. This finding provided the first molecular insight into the molecular regulation of tissue eosinophils. In order to prove this theory, we subsequently knocked out the eotaxin gene in mice. The results demonstrated an essential homing signal for tissue eosinophils. As the primary reservoir of eosinophils, the intestinal tract was nearly devoid of eosinophils. We went on to quantitate the level of eosinophils in the graph on the right. And please note that we published this graph without statistical analysis, no p-values. And this was based on Frank's motto, which was that if you need statistics, it's not real science. We went on to further prove this theory by demonstrating that eotaxin indeed had a role in regulating allergen-induced eosinophilia in the lung. We first showed that administration of eotaxin, which is in the background, and these are animated slides, unfortunately, but the image in the back is basically sheets of eosinophils that we found after we instilled eotaxin into the mouse lung. And we noticed its very rapid and eosinophil-specific activity, which was enhanced by IL-5 overproduction. Later, we had identified eotaxin-2 in mice, and we generated mice that were deficient in both eotaxin-1 and 2, and we showed in this graph that there was a 98% reduction in allergen-induced eosinophilia. After reviewing this data, Frank indicated that I'm now convinced that you have uncovered the molecular identity of a long-pursued eosinophil chemotactic factor of anaphylaxis that Frank had been working on with his colleagues for a number of years prior to these findings. A major focus of my research has been to understand eosinophilic esophagitis, not only because of its strong unmet need, but also because the disease is characterized by profound tissue eosinophilia. Furthermore, and importantly, the disease provided me with an opportunity to study human tissue eosinophils derived from routine endoscopic biopsies. Basically, EOE provided a bonanza source of human tissue eosinophils in the context of allergic inflammation. We have taken multiple approaches to understand esophageal eosinophilia, including a focus on the molecular analysis of esophageal eosinophils in murine models of EOE. Two recent approaches include single-cell sequencing, shown above, and ATAC sequencing, shown below. The latter approach, the assay for transposase accessible chromatin, permits a first-pass understanding of chromatin structure and accessibility in eosinophils. In particular, bar-coded transposases are integrated into accessible chromatin, and next-generation sequence is used to map the genome-wide epigenetic landscapes. This slide shows the data from single-cell RNA sequencing of the mouse esophagus following an EOE model. One can see that eosinophils are primarily composed of two populations based on this analysis, and these cells have been further characterized by the ATAC sequencing in the next slide. Panel A in this slide shows the complete separation of hematopoietic eosinophils, bone marrow derived, from tissue eosinophils based on their chromatin landscape. Panel B depicts this data in a heat diagram. These collective findings are quite striking because they show very strong separation between the eosinophil epigenetic landscape as a function of the eosinophil population. We use this information to uncover the potential functional role of eosinophils as shown in the next slide. Esophageal eosinophils are functionally enriched in pathways related to inflammation, which you can see in the circles, compared to the hematopoietic eosinophils. These eosinophils are very enriched in sensing response to external signals, TLR signaling, response to bacteria, response to LPS, for example, and apoptotic regulation consistent with the importance of survival in inflammatory sites. In contrast, bone marrow hematopoietic resident eosinophils are involved mainly in homeostatic metabolic processes. We use this epigenetic landscape information and RNA sequencing to map out the transcriptional factors involved in regulating tissue eosinophils, as shown here. In panel A, the predicted motifs are shown. Panel B highlights the enrichment of select transcription factors in esophageal eosinophils. Panel C shows the predicted interaction complex of the implicated transcription factors. And finally, in panel D, we show the ATAC peaks that share motifs for select transcription factors. What's shown particularly in these figures is that there is a remarkable conservation of an AP1 complex of transcription factors. Notably, these are uniquely different than the transcription factors that regulate eosinophil development. We next aim to prove that the role of tissue cues and select transcription factors in instructing the acquisition of tissue eosinophil phenotypes. In the upper panel, eosinophils were co-cultured with esophageal epithelial cells in the presence and absence of IL-13. This resulted in upregulation of relevant genes in eosinophils. Of course, these are just representative of many. This lower panel tests the role of specific transcription factors through collaborative studies with Annalise Pisalia and her mentor Kate Quinlan in Australia, where eosinophil cell line EO1 is knocked out for some of the transcription factors that are in the AP1 complex that we've identified. This includes JUN1 and ATF3, and the results of single knockout as well as double knockout are shown. And one can see that JUN has a generally positive effect, whereas AT3 is mainly a transcriptional repressor in eosinophils, and the knockout of both reveals cooperativity. So I'd like to conclude today in terms of the research that I presented with you with an overview of eosinophil development and tissue specialization. Our studies have identified IL-5 as a key developmental cytokine and eotaxin as a chemoattractant involved in eosinophil homing during homeostasis and inflammation. Furthermore, I have shared the concept that the tissue eosinophil is instructed by local environmental cues operating through distinct epigenetic and transcriptional programs, which are distinct from those involved in eosinophil development. I'd like to thank all the people that contributed to this work, which involves a group of diverse researchers coming together, including GI experts, research lab people, allergists, pathology experts, geneticists, and other investigators, as well as support groups, particularly the Curative Foundation, the SIGA group collectively, which is supported by the NIH, as well as the various NIH institutions and family foundations that have supported this work. I'd like to close by thanking K. Frank Austen for accidentally introducing me to the eosinophil, for his long-term mentorship, for teaching me about high-quality science and for demonstrating, as John mentioned, the importance of family, for teaching me to live life to its fullest. And I will stop here, or I will break one of KFA's cardinal rules, which was, quote, to cut out the editorials and just give the data. Thank you very much for your attention today.
Mark, thank you very much. Question and answer. If anybody has questions, please do post them. There are two that have come in so far. First one, should we still say that EoE is an eosinophilic disease, or is anti-IL-5 antibodies do not improve symptoms in this disease? Good question for you, Mark.
Yes. The latest findings indicate that we should probably not consider EoE as an eosinophil-mediated disease, particularly in the patients that have that we're treating after that have enrolled in the clinical trials per se. Basically, what we've seen in repeated studies, and I would encourage you to take a look at our recent Lancet GI paper, as well as the release of the recent Messina data, which is primarily in abstract form. But basically, eosinophil depletion with benralizumab had a very dramatic effect in eliminating eosinophils as expected, but had no effect on any other manifestation of the disease except for eosinophil-associated histopathology. This is including randomized phase 3 trial and also includes extensive transcriptomic analysis, which showed very little changes except for eosinophil signature genes. So these are extraordinarily surprising results, but very definitive results that focus our attention away from the eosinophil and perhaps to the mast cell, which is part of the reason that we're here today. And it doesn't fully rule out that eosinophils are involved, and certainly in the mouse system we're seeing alternative roles, but I think more data is still needed. But I would answer the question by saying, to first approximation, eosinophils are not the primary causal cell in patients that have EoE, typical of the ones that are enrolling in studies, which I'll just emphasize are people that generally are refractory to other therapy, have long-standing disease, and it does not rule out the role of the eosinophil in earlier stages of the disease, such as the development of EoE. Anyhow, I think I've answered enough on a very complicated question, but I'd be happy to talk more about it. It's certainly a very interesting question and important for the field.
Another question. Why have eotaxin-targeted antagonists and biologics not been pursued or been successful to date in eosinophil-associated diseases, asthma, EoE, others?
Well, I think that the answer to that question is somewhat similar to the other question, probably because of redundancy in the chemokine system. As you know, there are very few chemokine antagonists that actually have made it through the clinic, except for mainly infectious diseases, where the receptors are particularly important in select diseases like HIV. In any event, we even know now that eosinophils are eotaxins eosinophil selective because they're relatively selective for the eotaxin receptor, which is specific to eosinophils by and large. There are other cells, but that express CCR3, the receptor. However, CCR3 receptor is a promiscuous receptor. So eotaxins are not the only ligands. As a result, there is even at the level of CCR3 there is promiscuity. In addition, eosinophils express other chemoattractant receptors, including the classic ones, including C3A, which is actually something that's come about out of our genetic data, as well as platelet activating factor, leukotriene receptors. And so the basic answer is that the eotaxins in humans do not appear to be required, and it is a redundant system, whereas in the mouse systems, we've been able to map out into definitive roles in different experimental conditions. And the data that I showed you today with the eotaxin 1,2 double knockout has been as well as is reinforced by knocking out CCR3 in mice, where we see a very dramatic reduction in eosinophils as well, and different changes in the cardinal features of asthma in mice.
Does eotaxin have any effect on mast cells?
Generally not. No, CCR3 is not a mast cell chemokine receptor.
Marc, have you added mast cells to the esophageal cell eosinophilic co-cultures to ascertain any reciprocal transcriptional effects on the EOs versus the mast cells?
We haven't done that experiment yet. Certainly sounds very interesting. I'll have to do that with Francesca. Put the allergy unit on top of an ex vivo organ transplant or something like that. Thanks for that great question.
Great talk. Would you have any insights on mast cell eosinophil interaction in EOE?
I'm not sure about the interaction, but but we do have substantial interest in the mast cell being involved in the disease. We do know from our single cell data that there's a number of potential interactions that are quite interesting. One of those cells that express the highest levels of the histamine receptor 4, for example, is the eosinophil. Based on the single cell data, we see very high relative expression. Histamine obviously made by the mast cell primarily, basophils secondarily, but they're not heavily infiltrating in the EOE. So there's a number of similar reciprocal relationships, probably similar to what Francesca talked about in terms of the allergy unit that we've been able to see, and only time will determine the interaction. However, I do want to indicate that the benralizumab data, which is basically eosinophil depletion in humans, did not show any dramatic effects on mast cell signature responses based on the transcriptome analysis. For example, like CPA3 is a marker, a very selective marker for eosinophils, is not reduced by is not actually reduced, by benralizumab as a secondary consequence. So basically there's no evidence so far in human EOE that there's a reciprocal interaction that does not rule out a very dominant role for mast cells, which is certainly a hypothesis that's ongoing and being tested, particularly with the anti-c-KIT antibody by the Celldex company that's running a clinical trial now to test that hypothesis.
Marc, thank you very much. A number of questions, a very hot topic, so we very much appreciate your contributions. And for the sake of time, we will move on, and it's my pleasure to introduce Jenny Hallgren Martinsson. Jenny is an associate professor at Uppsala University and a senior lecturer on immunology. She defended her thesis on mast cell tryptase in 2004 under the supervision of Gunnar Piljer, and did her postdoc at the Brigham and Women's Hospital and Harvard Medical School, working with Mike Gersh and Frank Austen. Jenny has led her own research group at Uppsala since 2008, studying the role of mast cells and their progenitors in lung diseases, such as allergic asthma. Since 2015, she's been the secretary of the EMBRN Council, and she will be presenting on the topic of inflammation-induced mast cells in the lung. Jenny, take it away.
Okay. Hello, everybody. Can you hear me?
Yes.
Okay, let me see. Okay, so I'm very happy and honored to be able to give a presentation at this webinar, and in this way express my deep gratitude to Frank, and also to acknowledge his major contribution as a mentor to me and many others, and also to the mast cell field. And to me, the time in Boston really had a huge impact on my future research path. So when I finished my PhD, I very fast moved to Boston and joined Mike Gersh's group in Frank Austen's lab. And this was in January of 2005. And I came to stay in Boston for almost 3 years. My postdoc project was based on the fact that already in the 1990s, it has been discovered that in patients with asthma, mast cells are not only activated and release mediators, but they also accumulate at specific sites of the lung, such as in the airway epithelium, and also later it was shown in the smooth muscle cell area. So my postdoc project became to use an OVA model of allergic airway inflammation in mice to look at the recruitment of mast cell progenitors to the lung. We used different knockout mice, and we used blocking antibodies and so on, and tried to describe the mechanism behind this recruitment response. And at this time, to quantify the mast cell progenitors, we used a very time-consuming limiting dilution assay. However, when I returned to Uppsala and eventually had the opportunity to start my own group, I wanted to use flow cytometry to be able to really look at these rare cells more specifically. So I want to introduce first a little bit about what we know about mouse mast cell progenitors. So these are hypogranular cells that, just as their mature counterparts, show surface expression of c-KIT and Fc-epsilon-R1. And already in the first study with Mike and Frank, we could show that these mast cell progenitors need to express alpha-4 beta-1 and alpha-4 beta-7 integrins in order to become recruited into the inflamed lung. These cells are extremely rare in homeostatic conditions, and when we quantify them in tissue of naive mice, they correspond to about 50 cells per million isolated mononuclear cells. Later, we have shown that not only allergic inflammation, but also influenza infection can trigger the recruitment of mast cell progenitors to the lung. And this means that they expand in number about 50 times, so they are still quite rare, especially in comparison to eosinophils. You can also use more specific triggers like intranasal IL-33 or poly-IC, and this can also expand the number of mast cell progenitors in the lung. So a few years ago, Erica joined my group as a postdoc, and she set up the house dust mite model of allergic airway inflammation. And at this point, we had learned how to use flow cytometry to quantify these cells. And here you can see first example of a PBS injected mouse, or it could be a naive mouse as well, because they have very few mast cell progenitors and mature mast cells. And we can use the level of beta-7 integrin to differentiate between them. Here you can see an example of an HDM-treated mouse, and you can see that the mast cell progenitor population has expanded a lot. You can also see that there is a third population here with intermediate beta-7 integrin expression, and we call them the induced mast cells. And we have used this model in a number of papers, but today I want to show you something that is unpublished. So a few years back, Abby joined the group, Abigail, I should say, as a PhD student, and she also used this model of allergic airway inflammation. Because in one of her projects, she wanted to look at what happens with these HDM-induced mast cells upon resolution of airway inflammation. We thought that this was a really relevant question to ask, since asthma is a recurrent disease that oscillates between acute allergic inflammation and then like a healing phase. So Abby looked at different time points after the last HDM administration and could see that after about 24 days, if she looked by histology, she could see that the macroscopic signs of inflammation were more or less gone, and the lung tissue looked like a naive mouse lung. So she used first the same panel as I showed you before to quantify mast cells by flow cytometry. So here you can see first that a The muscle progenitors will be, of course, more at acute allergic inflammation, but upon resolution, they will decline in number. The induced mast cells, they seem to stay similar in number, and the mature mast cells, they increased in number. So then I just want to remind you that in the mouse, we have two major subtypes of mast cells, the connective tissue type mast cells and the mucosal type mast cells. The connective tissue type mast cells, they have an embryonic origin, and they specifically express mouse mast cell protease 4. The mucosal type mast cells, they specifically express MMCP1, and at least in the lung, it seems like both these types express the tryptase MMCP6. So Abby used these proteases as markers for these two major subtypes, and I want to show you this picture as a representative, showing the lung day 1, that is 1 day after the last HTM challenge. You can see these beautiful mast cells that appear in the epithelium, and you can see that they co-express MMCP1 and MMCP6 and therefore become yellow here. However, interestingly, at day 24, when we had the inflammation had been resolved, we could see that the mast cells are still in the epithelium, but they have lost the expression of MMCP1. So Abby did a great job here looking at different sites of the lung and trying to quantify the number of mast cell protease positive cells. So now we are just looking again in the epithelium. Of course, in naive mice, you don't have any MMCP6 positive cells in the epithelium. However, with the mice with acute allergic airway inflammation, on day 1, there are mast cells there that stain positive for MMCP6, and it seems that they stay there at least until day 24 in similar numbers. When she quantified the mucosal mast cells using the MMCP1 as a marker, can again see that there are no MMCP1 positive cells in the naive mice. All mast cells that we can see day 1, they will stain positive for both MMCP6 and MMCP1. But you can see that the expression of MMCP1 gradually go down, or there are fewer cells that express MMCP1 when the inflammation starts to be resolved. And she also used integrin beta 7 as a marker for the mast cell progenitors, and could see that at acute allergic inflammation, many of the cells that line the epithelium are co-staining for integrin beta 7, suggesting that they are mast cell progenitors at this stage. And we can see also that like a week later, there are much fewer cells that stain positive for integrin beta 7. So this is just a few slides showing some data from a much larger study that we hope to be able to present in another forum soon. But so far we can conclude that it seems like in mice with acute allergic inflammation, the mast cell progenitors become recruited into the lung, and they go to the epithelium. They express MMCP1 and MMCP6, and they lose quite rapidly integrin beta 7 and become mucosal mast cells. However, upon the resolution phase, while they lose the MMCP1, they still stay there and express MMCP6. So we think this is kind of a sign of innate memory at this site. Okay, so what about humans and human mast cell progenitors? So in 2016, we published this paper with Joachim Dahlin as the lead author, and that was the main experimentalist in this study, where we could show that human blood also contained a similar population of mast cell progenitors. These are hypogranular cells. They have a typical progenitor type of morphology. They express c-KIT and Fc epsilon R1, and they are about the same. They are as rare as the master progenitors in the mouse. And they stain very weakly with the basic dyes. So you can see in comparison to lung mast cells and blood basophils that were stained on the same time. So what did we learn about the human ones so far? Well, for example, we have seen that these cells can be activated by IgE cross-linking, and this is shown here looking at or using a cross-linking anti-IgE antibody, showing that the activation marker CD63 and LAMP1 is higher in those cells that were activated compared to those that were only treated with vehicle. We have also tried to ask and answer the question, what does it mean to have these circulating master progenitors in the blood? And Maja Salomonsson, another previous PhD student, she analyzed 38 patients with well-controlled allergic asthma, and she could see that those patients that had a reduced lung function, they also had a higher master progenitor frequency. In a study that was one of Abigail's PhD projects, she also analyzed birch pollen sensitized asthma patients in birch pollen season, which occurs in Uppsala between April to early June, and out of pollen season in November to January. And in this study, we could see that the master progenitor frequency was higher in birch pollen season in May compared to November. And looking at the patients that was exposed to the birch pollen, we could see that those that had the highest frequency of master progenitors, they also had a reduced asthma control and more asthma and allergy symptoms. So to conclude, I think that we can say that master progenitors in mouse and human show many similarities, and probably it's valid to use mouse as a model to learn more about the mast cell biology in the context of disease. I think it's very interesting to see this, that the natural allergen exposure increased the blood muscle progenitors temporarily, and that the occurrence of these progenitors in the blood seem to be related to reduced asthma control and more symptoms. And there are, of course, many outstanding questions here. I also want to acknowledge Mike and Frank, especially, for the inspirational time that I had as a postdoc in Boston, and that really affected my future thinking about mast cell biology, I would say. I also want to thank all the amazing people that have been in my group, both the past PhD students and postdocs, and the present group they are really fantastic. And I also want to thank our recent collaborators and funders. Thank you very much.
Jenny, thank you very much. Are there any questions for Jenny? I think if you have very nice talk. I'm sorry. Very nice talk. ILC2s become memory-like after HDM and produce elevated and different cytokines. Are the elevated MCP6+ mast cells after resolution? ILC2 dependent. Good question.
Okay. Yeah, I have to say that I don't know that this is very new data that we have also, but yeah, no, I cannot really say anything about that.
Okay, thank you. I think because of time, we'll move on. I would like to have afford an opportunity to Francesca to show her final 2 slides if they're available.
Thank you. I need the share screen. Yes.
And I've
Yes, I apologize with all of you for something happened and I couldn't show to you my last 2 slides. One is, of course, the thanks to you, to all my students and past and the present collaborators. And the last slide is that I would like to share the poster of our 139 kidnapped civilians in Gaza that are now for 112 days there. And we have also a 1-year-old and a 3-year-old toddler. And I hope you pray with me for their safe return, for peace in the area, for both the Israeli civilians and the Palestinian civilians. Let's this horrible war stop. Thank you. And thank you, Peter, for letting me show these 2 slides that were cut off. Thank you.
I think it's notable that, all of our presenters today were trainees with Frank Austen. And they in turn are training the next generation of investigators in the broad fields related to eosinophils and mast cells. So the legacy of Frank that is well recognized and captured in some of the presentations this morning. There were other areas of immunology that Frank also had contributions to. Complement. I met my wife in Frank's lab. She was a complementologist. So I have to acknowledge that there's more to science than eosinophils and basophils. But I think that this webinar is really a testimony and a recognition of the enormous impact that Frank had in the fields of science and on the training of the future of current generation and future generation of investigators and physician scientists. So with that, I remind you that this webinar has been recorded. It is available on demand. We will be querying you with an evaluation of this webinar, so that will help inform us as we go forward with future webinars. So again, I thank all of our contributors today. I thank the 2 societies that are sponsoring this webinar, the International Eosinophil Society and the European Mast Cell and Basophil Research Network. So thank you to all who presented. Thank you to all who joined this webinar and wish you all well. Thank you.